Okay? So remember, a hit is only constituted when radiation inactivates that target molecule. So, when the radiation dose reaches a level sufficient enough to kill 63% of the cells, this is the funny, this is going to be flipped around. With a 37% survival, this is known as D37, because it's based on the number of cells that survived. Not how many died, but on survival. D37 refers to the amount of radiation necessary to kill 67% of the population with 63% of the population with 37% of survival. Okay? So, in terms of radiosensitivity, D37 is a measurement also of radiosensitivity of the cell. If you have a low D37, that means, remember it says up here, the amount of dose necessary to kill 63% and survive 37%. So a low D37, you don't need a lot of radiation. It's low. I'm talking about the dose necessary to get 63 and 37. The dose is low. So therefore, that general population or colony of bacteria is radiosensitive. If you have a high D37, now we're talking about we need a higher dose to reach this 63% death and 37% survival. This type of colony or group of bacteria is radio-resistant. We need more. So high D37 means we need more radiation. Low D37 means we need less radiation. We don't need as much. Okay? I'm sorry? Oh. It was. It was, okay.
Sorry.
Okay. All right, so low D37 is you very sensitive, you don't need a lot of radiation. High D37 means you need a lot of radiation. Okay? Make sense? Yeah? Okay. And then this just sums it up. If it wasn't random and if it was selective, if you had 100 raindrops and 100 tiles, the D37 is the dose that would be sufficient, with no wasted hit. D37 is the dose that would be sufficient in killing 100 of the cells. All right. Let's talk about the multi-target single hit model. That refers to us, the complex organisms. What it's saying here is, with humans, we have multiple targets. So you got to hit those targets before the cell is considered a hit. Okay? So going back to Poisson's law of random distribution again. Now each tile is going to be divided in half now. Okay? Each tile is going to be divided in half. For it to be considered wet, you have to hit both those halves. Okay? So each square is divided into two more equal parts. A raindrop must fall on each part of the square to be considered wet. Otherwise, it remains dry. With little rain, okay, again, compare this with radiation exposure. With little rain, no squares may be wet. However, when you increase the raindrops, you're increasing the chances of raindrops falling on those two pieces of the tile. But as long as it stays within that, if you're only hitting one and not two, that's what's known as the threshold. And if you stay below that, this is where cell survival occurs. You guys follow that? Okay? Because again, multi-hit means you got to hit those different targets for the cell to die. If you don't hit all those targets, you're giving the cell an opportunity to survive and recover. Okay?
So you don't
That's why we can do what we do. That's why we can do what we do. Because when you're in a room shooting x-rays and doing fluoroscopy, yes, we're getting hit with radiation. Okay? But because it's random and because we're multi-target, It may be interacting with our cells, but it's not hitting all the target cells, and our cells survive and they can recover. So with increasing number of raindrops, eventually an entire square will get wet. But that's the key, is you need to increase the raindrops or increase the dose until a hit is commissioned. Complex biological specimens like human cells are thought to have more than a single critical target. So if a human cell was to have 2 targets, both would have to be hit in order for the cell to be inactivated and die. With very low radiation doses, cell survival is nearly 100%. However, as the dose increases, so does the chances of getting hits on a target molecule. Okay. Again, compare it with the raindrops. If we're increasing more raindrops, there's a greater chance for those tiles to get wet. If we increase radiation, there's a greater chance for those multi-targets to get hit. At even higher doses, most cells that survive may have been hit once. Therefore, at this point, it's now going to follow the single target hit theory. Does that make sense? Because it's already been hit once. Now it's going to flip over the single hit theory, because all it needs now is to get hit one more time before the cell dies. Okay? So once hit, and a multi-target theory, once hit, it now follows the single target, single hit.
So the last one, you said that if it hit one, the cell dies. So if it hit again, it doesn't do anything, right? The last
For which one, the bacterial or the multi-target? Are we talking about single hit or multi-target?
Oh, I think the multi-target.
Yeah?
Yeah, the bacterium.
Yeah, bacterial, yeah. So bacterial just hit it once, it's dead.
Okay.
That's a single hit. But we're more complex, so we have multiple targets.
Okay.
Yeah, so it's got to be hit more than one time.
Okay.
Okay, so remember the D37, right? D37 refers to the amount of radiation needed to kill 63% of the population of the cells, and then 37 survival rate, right? Okay. In the multi-hit target, it is DO, or D0. It's the same thing, but D0 applies to the multi-target single-hit model. And just like the D37, a high DO indicates cells that are highly radioresistant, whereas a low DO indicates radiosensitivity, so you don't need a lot of radiation. Now, if you look at the very top over here, notice again, it's the straight line is what's directly proportional, okay? So with increased amount of radiation, more cells are dying, right? Up here, low doses, it's not affecting so many cells, but as you increase your dose, more cells die, okay? You will notice that there is a curve here. When you see a curve, does that mean proportionality? Is there direct proportionality when you see a curve? No, you don't. It's only the straight portion that you see proportionality. But here it's curved. So what it's saying here is at the lower levels, yes, there is some response, but it's not an immediate response as the dose increases. It's not until you pass this portion here that it now becomes directly proportional.
So it's like its threshold?
Yes. So what this DQ is, I'm thinking Dairy Queen, okay? What DQ is here, it refers to the threshold. So as long as you stay within that threshold, there is a chance for cell survival and cell recovery. Now you go beyond that, okay? Now you get cellular death. Okay? So DQ refers to the threshold. Refers to the threshold dose or the capacity of the cell to recover from a sublethal dose. This represents by a curved shoulder instead of a straight line. Now, if the curve is wider, the more sublethal damage it can sustain. when doses are delivered sublethally, cells that survive may recover and exhibit the same characteristics as when it was originally irradiated. Okay, so again, if it was done sublethally, the cells will behave in the same manner as they did prior to being irradiated, is what it's saying. Back in a normal state, normal behavior. Okay, any questions?
Sub.
Huh?
Sub.
Sub.
Oh, I get it.
Sub, yeah, below. Below lethal dose. Sublethally. Okay. Let's talk about cell cycle effects. Cell cycle time. Cell cycle time is cell generation time. It refers to the cell's replication in mitosis from one mitosis to another. Most human cells, their cell cycle time is 24 hours. Specialized cells takes a little bit more longer to replicate. Cells like neurons do not replicate. Regeneration time varies. Variations occur at the G1 stage of interphase. Cells in mitosis are always radiosensitive, right? Because that's at the time right before they split. Okay, so you don't want to irradiate them right before mitosis, because if you cause cellular damage, especially at the DNA, you're going to carry that damage code to the daughter genes, daughter cells. Okay? So they're especially radiosensitive in the G1 to S transition and less radiosensitive in the late S. Okay. Talking about interface in typical terms. Okay. LET, RBE, and OER. I had to get, this concept is very difficult to understand, so I had to get pictures which will demonstrate what I'm trying to convey here, okay? So just bear with me. I'm not into war, I'm not into violence, okay? So high LET refers to substances or particles that are very, very thick, very dense, and they're not highly penetrating, such as alpha particles and neutrons. So high LET follow the single target, single hit model, which means that when you have something that's this huge and can cause a lot of power, all it takes is just one hit to cause the damage to that wall. Okay? So all you need with high LET particles is you just need one of these particles to cause this much damage to the cell. Is that making sense? Mm-hmm. Okay? Now if we refer to the low LET, like X-rays, you're going to have to, you're going to have to hit this wall many, many times before this wall is going to come down. Okay? So this follows, low LET follows the multi-target single hit model. So you got to hit this multiple times before you can cause some serious damage. Not like the wrecking ball. I was going to put Miley Cyrus on there, but I didn't think I Okay? So the mean lethal dose for low LET irradiation is greater than that of high LET.
Is that like those little bullets?
These little holes right here? Yeah, those are the hits caused by that.
But they're more powerful than the ones they hit? They're just not as small?
No, they're not as powerful because they don't transfer as much energy as something that is high LET. High LET is releasing a lot of energy.
But
And
that
this,
doesn't mean lethal dose greater than the high.
okay, which slide are we talking about?
It's how is a low LET greater
The next
than that one that we have right there? That's what it kind of seems like it's saying.
Okay, this one over here?
Uh-huh.
Okay.
Bottom.
The mean lethal dose for low LET radiation is greater than that of high LET. The mean lethal dose. The average lethal dose for low LET irradiation is greater than that of high LET. Yeah, so you're going to need more of this. What it's saying here is you're going to need more of low LET to create the same kind of damage as someone that has high LET. Yeah? Sounds good? Yeah, so you need more. So you need more of this to cause the same kind of damage than one that has high LET. You don't need a lot of that to cause the same amount of damage. This you need more. Yeah.
You get it? You get it?
I get it.
Okay.
I don't know how else.
That's what she said.
If you
Yeah.
guys can rephrase it a different way, help me out.
It's the same way with how you
You know,
explained
you said
earlier.
average was better.
Yeah?
Average is better.
Average is better? Okay, average, okay. All right, and then we know about oxygen. Oxygen is the most studied dose modifier. It maximizes the effect of low LET radiation. Again, we don't have to use alpha or those different type of particles. We can use X-rays because we're depending on those X-rays to interact with the water molecules to cause free radicals. Okay? Any questions? Okay, let's cover one more chapter.